In the last section, we talked about this idea of preventing things from getting into the body. Now we're going to talk about the third line of defense, which is specific to particular viruses or bacteria. These are things you've been sick with before. Your body learns how to recognize them and prevent getting them sick a second time. Specific defenses protect us against specific pathogens where non-specific defenses fail. It also helps protect us against cancer. And it depends on the action of two very important cells. These are your B and T cells. These are types of lymphocytes or white blood cells. B cells produce plasma cells and memory cells. Plasma cells are what actually produce your antibodies. These are what your body is bombarded with once you've learned how to fight off a disease. Memory cells are kept around so that if you are ever invaded again by this virus in the future, you can prevent yourself from getting sick. So, for example, if you had one particular type of flu, you can then create these antibodies to fight that flu off. If you were exposed to that one specific flu again, the memory cells would then flood the system with antibodies and you would stave off a second infection. T cells regulate the immune response. They produce various types of T cells. They're the cytotoxic T cells, which kill viruses infected in cancer cells, the helper T cells, which regulate immunity. Memory T cells are ready to kill in the future. So you can see the differentiation here between the types of B and T cells. Here you could see the antibody-mediated immunity, where you can see the B cells being stored both as memory cells with those antibodies on the outside. And then here you can see the plasma cells that are pumping out these antibodies that are then exactly the right shape to fight off that exact particular virus. Notice how tiny those viruses are and how tiny those antibodies are. You can also see the T cells, which are helping mediate this response. Here is a cell that's been infected by a virus. It's presenting that antigen or the virus, and then the T cells are attempting to find the correct shape of antibody in order to be able to fight this off. Once it does find it, it will create this cascading response that leads to all of those antibodies being produced. So characteristics of your B cells, they're antibody-mediated immunity against your pathogens, meaning they're producing antibodies. They're produced and mature in your bone marrow. They recognize a very specific shape of antigen and undergo clonal selection, meaning once they find the right shape, they then start pumping out millions and millions and millions of these antibodies that are the right shape. Clonal expansion produces antibody-secreting plasma cells as well as memory B cells. So let's talk about antibody-mediated immunity. Each B cell has a unique receptor called a BCR. It binds to a very specific shape, one particular antigen. So perhaps it's the virus that causes chickenpox or the virus that causes measles. Whatever that is, it's got a very specific shape, and we're going to call that an antigen. And when that B cell has been exposed to the antigen, it will eventually, it takes about 3 days, find the correct shape to be able to fight that antigen. Binding and cytokines secreted by helper T cells result in clonal expansion. The B cell makes lots of copies of itself. So we've got this interesting shape that's new. The B cells work to find the right shape. Once they do, they pump out millions and millions of copies of itself. Most of the cells that are produced are your plasma cells. These are what are secreting your antibodies. The other cells that are produced are your memory cells, which go into your long-term immunity. Sometimes if you're not exposed to something for a really, really long time, you will actually need to be exposed in a little bit of quantity in order to boost your immunity again. This is why some vaccines have to be administered again after a few years. After an infection has passed, the plasma cells undergo this programmed death, leaving only the memory cells. So here you can see this antibody-mediated immunity by the B cells. There's the antigens. Notice they have a very specific shape. We have these B cells that are making all of these different shapes, attempting to get the one that will actually work. If you get really lucky, sometimes you'll get it within a matter of just a few hours. But on average, it's going to take about 3 days in order for you to get the correct shape. Once you do, it's going to cause this chain reaction that is going to cause the formation of plasma cells to pump the antibodies out, but also a few memory cells to be hung around. So, in case you encounter this same virus again, you can then make those antibodies again. Once the infection has been resolved, you don't need all of those plasma cells anymore, and you have this programmed cell death, leading to just the memory cells being kept around. So when we want to think about what is the structure of an antibody, the basic unit that composes an antibody molecule is a Y-shaped protein. The trunk of a Y is a constant region that determines the class of the antibody. The ends, or your arms, are the variable regions where specific antigens can bind. These are the parts where different proteins are used to try to make different shapes. Here you can see those binding sites at the end of the Y, and it's got to be exactly the right shape and the right size for this specific antigen. Each disease, or I should say infectious disease, typically will have a very specific antigen for it. So you need a different antibody for each one. So here's just some of the different classes of antibodies. I have some homework questions about this so you can play around with them, but I don't expect you to memorize these for your test. So that was B cells. So now let's talk about the characteristics of T cells. Cell-mediated immunity against virus-infected cells and cancer cells. Oh my goodness, what does that mean? Basically, what this is saying is if you have a normal cell and something's wrong with it, maybe a virus has infected it or it's turned into cancer. Typically, when it turns into cancer, some other things have gone wonky. What we will find is that T cells play a really big role in fixing this. So they're produced in the bone marrow, they mature in the thymus. An antigen, remember that's the bad stuff, must be presented in a groove of this one particular molecule. The cytotoxic T cells destroy any non-self antigen-bearing cells, meaning if you've got a cell and it's starting to get invaded by a virus, those T cells can identify that there is a pathogen in there or an antigen, and they'll destroy that cell, and they'll also take hold of that pathogen. And we'll talk about what that's going to do in a minute. And the helper T cells then secrete cytokines that control the immune response. So, again, this is part of our specific lines of defense. It's cell-mediated immunity by the T cells. So each T cell has a unique receptor that's called a TCR. It recognizes a piece of an antigen with the help of an APC. This is the antigen-presenting cell, which is a really fancy way of basically it goes, hey guys, I'm me. They have a little protein that sticks off the end of them that says, I'm me. And then they take the antigen, and after they've engulfed it, they break it down and move it to the surface and put it right next to the part that says, I'm me, the human leukocyte antigens. And that way, the body knows this is a good cell and it's helping me figure out what the bad thing is. And it goes to either your lymph nodes or your spleen, and it presents both the I'm me and the bad antigen side by side. So, because they can have these two things side by side, it allows these helper T cells to start finding the correct shape of antibody. It's leading mostly to the helper T cells, the cytotoxic T cells, and a few memory T cells. So after an infection is passed, the helper and cytotoxic T cells undergo that planned destruction, leaving only memory cells again. You'll notice this sounds a lot like what happened with the B cells. There's redundancy. And the reason is because this is how we keep from dying of disease. Our immune systems are fan-freaking-tastic. We are constantly bombarded by pathogens and viruses, and our immune system takes care of almost all of them without us even having to notice what it's doing. And it's because it was so powerful that we have a lot of these redundancies built into the system. So here you can see one of these guys that happens to have that self antigen along with the bad guy, that other antigen right here. So now you can see that we've got the cytokine response. We're seeing the correct activation of the T cells, of finding the correct shape. And then you can see those memory cells, and when they're done being used, they destroy themselves. So helper T cells secrete the cytokines that help many immune systems function. These are the things that start ramping up the immune response. These cytotoxic T cells that have the vacuoles containing these proteins that I'm not concerned that you necessarily know, but they are basically what we need to have. These are those different proteins that are going to help us. These perforins are what's punching the holes in our target cells, followed by these other proteins that cause the cell